Chapter 7 is all about the membrane and transport across the membrane. In this segment, we're just going to talk about the membrane in general, its structure, its fluidity, and things that can affect its fluidity. And then we'll move on to transport across this membrane. So this main picture shows some space filling model diagrams and some stick model diagrams of a combination of things, right? So we have a bunch of water, and then we have the polar heads of the phospholipids that make up the plasma membrane, and these would be the hydrocarbon tails. The blue ribbon-like diagram represents a protein that looks like it spans across the membrane, and then you can see some water molecules kind of crossing that I guess within membrane, or the plasma membrane, is basically a boundary between the inside of the cell and the surroundings of the cell, or the outside of the cell. It separates a cell from its surroundings, right? And it kind of protects it, and it serves as a gate, right? So it just it only allows certain things in and certain things out. So as I stated, the membrane serves as a gate. Okay, so it only allows certain things in and certain things out of the cell. This makes it what's called selectively permeable. Permeable meaning allowing things to cross it, right? Or allowing things in and out. And it's selective, meaning only certain things can go in and certain things come out, or certain amounts of things. Transport proteins are usually the gatekeepers of that gate, right? So they're there, and they are the ones that decide what goes in and what goes out. One example is this transport protein here. So you can imagine that we're looking down into, let's say we're looking into the cell. So all of this is plasma membrane right here, and then we're looking down through this channel protein. This channel protein is only big enough, the space there, the channel, is only big enough to allow the potassium ion, right? So a lot of times so the potassium ion is positively charged. So there's going to be a certain charge differential in this particular space right here, or the channel, which allows this positively charged, this size positively charged ion through that channel in and out of the cell as needed. Okay, so aquaporins are an example of a channel type protein, and it's specifically made for water. So you can kind of see this shows an aquaporin right here, and then of course the water molecules are moving across the membrane into the cell and or out of the cell as needed. Now, going back to the structure of the plasma membrane, if you remember the plasma membrane, I think we kind of introduced it when we were talking about the phospholipids. It's made up of phospholipids, and it's called a lipid bilayer, and it forms this bilayer readily when it's put in water. Because remember, phospholipids have the hydrophobic tails and then the polar heads. Of course, the hydrophilic or polar heads like to interact with water, so then the hydrophobic tails are going to try to hide from water. So it creates this lipid bilayer to hide the hydrophobic tails from the water. And you can see these space filling models here of the plasma membrane. Now, there was a couple models introduced in terms of the structure of the plasma membrane, and one final model would be the fluid mosaic model. And the fluid mosaic model states that the membrane, the plasma membrane, is made up of phospholipids with protein molecules embedded in it in a varying distribution. Now, there's a number of things associated with the plasma membrane. I'm going to kind of show you these things right here. So here you can see this is the plasma membrane here. And then we've got these purple things that span the membrane. And then we've got purple things that do not span the membrane. And then we've got a number of things outside of the cell which kind of attach to the membrane in some way. So let's talk about some of these things. So you remember the microfilaments of the cytoskeleton inside the cell. Okay, so again, those serve many purposes. But here we're talking about the proteins that are embedded in this membrane. So those that are embedded in the membrane and touching this hydrophobic portion of the membrane are called integral proteins. Those that do not embed in the membrane, those are more on the surface of the membrane or maybe on the surface of a protein that is an integral protein. These are called peripheral proteins. Now, integral proteins can span both sides of the membrane, or they might not go quite as far. Okay, they're both integral proteins, but those that span both sides of the membrane are called transmembrane proteins. So they're transmembrane proteins. Now, we're going to talk about this a little bit further, but I want to mention that cholesterol is important. You always hear these bad things about cholesterol and how you should limit your cholesterol. Yes, you should limit the cholesterol. However, you do need some cholesterol, and your body makes some of it. It gets incorporated into the membrane, and it helps regulate the fluidity of the membrane, and we'll talk about that later. Let's see, we've got a lipid here which has carbohydrates attached to it. This is called a glycolipid. So whenever you see this term glyco, that means it's referring to carbohydrates. So here, this is a carbohydrate lipid. That means it's a lipid that's, in this case, part of the plasma membrane that has carbohydrates attached to it. And then likewise, similarly, we have proteins that can have carbohydrates attached to them as well. Those are called glycoproteins. And then there's a matrix that's outside of the cell. In many cells, not all of them, but especially in animal cells, we have what's called the extracellular matrix. The extracellular matrix are made up of fibers of mainly protein and carbohydrates, which is connected to the plasma membrane, and it allows some communication and some stability to the plasma membrane. All right, so let's talk about the fluidity of the membranes. Now, membranes are basically held together weakly by hydrophobic interactions, because remember, we're dealing with hydrophobic molecules, while some of them might be slightly hydrophilic, as in the phospholipids. Those are actually amphipathic. However, there's a lot more hydrophobic interactions than there are hydrophilic interactions that actually hold the membrane together. Now, most of the lipids and some of the proteins can move within the membrane. So they can move side to side. So they can move from one place to another in the membrane. Now, some lipids may flip-flop or cross the membrane. So they might go from one side of the membrane, which might be the extracellular side, to the intracellular side of the membrane. This is quite rare, and it takes a little bit of energy to be able to do this. Now, let's talk about an experiment that is described in your textbook, which kind of showed that the lipids can move side to side. So we have this right here. We have a mouse cell and a human cell. Okay, so they're two separate cells where the membrane proteins were labeled with a fluorescent molecule, right? Different colors. So here the mouse cell looks pink, right? So it's got a pink fluorescent molecule on it. The human cell looks like it's got a purple or blue fluorescent molecule on it. Well, then they fused those cells together. So they fused together to create a hybrid cell. So now we've got one cell, one larger cell, which is part mouse and part human. Okay, with two different labels. So initially, look, at time 0, one side of the cell is going to be purple or blue, and the other side of the cell is going to be red or pink. Let's say after an hour, look at. Basically, those proteins were redistributed along the surface of the cell. So you can see that the proteins, lipids as well, can move side to side along the membrane. Okay, so let's talk about cholesterol. An animal cell helps regulate the fluidity of the membrane. So if we look at this right here, we have, remember the difference between saturated and unsaturated fatty acids. So saturated fatty acids do not have the kinks in them, and unsaturated fatty acids do have the kinks. So here, let's look to the right, and we've got this, this membrane, let's say it's an animal cell membrane, where it's made up of phospholipids that have saturated fatty acid tails. This makes it very rigid, right? Here it's calling it viscous, right? So very thick. It's very rigid. Now, if we look at opposed to that, we have fatty acids that have that is saturated, and you can see that they have cis double bonds within them, okay, because there are kinks. Now, the kinks create space in between the fatty acids, which make it more fluid, right? So it makes it loose, more loose. Now, temperature can change this situation, right? So as the temperature cools, a membrane that has more saturated fatty acids is going to become too rigid. One that has a fluid, more unsaturated fatty acids in there will be okay when the temperature cools. Now, when the temperature heats up, right? So when it gets hot, this fluid one is going to be too loose. It could fall apart. And the viscous will be okay. So it'll make it a little bit more fluid, right? So a lower temperature is going to make the membrane more rigid. And a higher temperature is going to make the membrane more fluid. Now, when the temperature increases, this fluid one is going to have a problem, right? Because it's too loose. There's not enough packing to keep it together. So there needs to be more hydrophobic interactions in order to keep it from falling apart. Cholesterol is embedded in the membrane in order to do this. Okay. Now, cholesterol helps the plasma membrane in both situations, whether there are unsaturated fatty acids or saturated fatty acids. So here, let's look at a membrane where there's all saturated fatty acids. Remember, that becomes too rigid. Well, the cholesterol makes spaces in between them, so it kind of breaks them up a little bit. Okay, even though cholesterol is mainly hydrophobic, it still breaks up that tight packing that happens with those saturated fatty acids. Now, if you can imagine, I don't really have a good figure for this, but if you can imagine cholesterol embedding itself into those spaces with the fluid membrane, the more fluid one where there's unsaturated fatty acids, it could fill in those spaces and keep it together. Okay, so back to the temperature. Okay, so during warm temperatures, cholesterol it restrains movement, so it keeps the membrane from falling apart. It keeps it together. During cool temperatures, it keeps the membrane from being too rigid, so it makes it more fluid. Now, there is differences between organisms in terms of lipid composition. So there's special adaptations that evolved in certain organisms. So some organisms can vary the composition of lipid as an adaptation to their specific environmental conditions. For instance, okay, some organisms can increase their saturation, so that makes them more rigid. Okay, so let's say they live in extremely warm conditions and they want it more, they want more saturation to keep or they can handle more saturation to keep their membranes together. Some of them can decrease their saturation, so this means this increases the unsaturated fatty acids, which makes it more rigid. So let's say the temperature is too cold, so it suddenly becomes cold. An organism can increase the unsaturation or decrease the saturation to keep it from being too rigid, right? So it makes it more fluid. Some of them can increase the length of their fatty acids, which makes it more rigid. And some of them can decrease the length, which creates more space, which makes it more fluid. So some organisms can adapt to their environment by changing the lipid compositions of their membranes. Now, let's talk about these proteins again before we move on to the rest of the membrane and move into transport. So I kind of already mentioned this when we looked at the overall picture, but you can see here we've got the alpha helices of this particular protein, and we've got a protein that spans both sides of the membrane. So this is the extracellular side, so this is outside of the cell. And then we have the intracellular side, or the cytoplasmic side of the membrane. So we can see this protein spans both sides. It's actually a transmembrane protein. It's an integral protein, but it's specifically a transmembrane protein. Now, transport across the membrane involves these types of proteins. So some of them require the energy from the hydrolysis of ATP. Some of them just make a channel of a specific size to only let certain molecules of that size in. Some of them act as enzymes. Okay, so some of them at the membrane can help catalyze chemical reactions. Some of them can act as a receptor, right? So we could have a receptor where you have a specific molecule that binds to that receptor and then induces a bunch of chemical reactions that happen inside the cell. Some of them can serve as recognition of cells. So here, look, we've got a protein with carbohydrates attached to it. Well, these carbohydrates fit perfectly into this region of another protein. So these two different cells are recognizing one another via these carbohydrates that are attached to the protein. And this is, again, a glycoprotein because it has those carbohydrates attached to it. Some of them can just serve as, protein-protein interactions to join cells together. And then some of them, of course, can, as we saw before, can attach the extracellular matrix to the proteins of the plasma membrane, keeping all of this connected. Let's talk about something really, really cool. This is kind of related to protein-protein interactions. So everybody kind of knows what HIV is. It's an infection. It's a virus, right? So it represents a HIV particle. Okay, so it's a viron is what it's actually called. And then it's got a genome. Well, it's got proteins on the outside of it, it's got a protein coat is what it's called. Well, this happens to be, let's say, a human cell, and there are receptors on the surface of our cells which allow HIV, the virus, to attach to and then basically fuse with the cell and incorporate its genome into the cell. So these two receptors are the following. We've got the main receptor, which is called CD4, and then it's got a co-receptor, which is CCR5. So both of those are actually required for HIV to enter the cell. Okay. Now, what's interesting is that a small percentage of the population, I don't know, maybe like 2% of the population, have a mutation in CCR5 so that they do not have a functional co-receptor. They cannot contract HIV. So they're basically, I mean, I hate to say the word immune, but HIV cannot enter the cells because this co-receptor is not functional. Okay, it's called delta 32 if you want to look it up. Delta 32 means that this CCR5 in those individuals are missing 32 amino acids. So it's cut off. All right, so it's quite interesting. So this kind of shows how important proteins interactions are when they're present and or when they're absent, right? So this kind of makes it good. I'm not sure what the effects are about other things regarding those individuals with CCR5. However, for this, it's good, right? So they have the ability to evade the HIV. Okay, so let's talk about carbohydrates and their role of cell-cell recognition. So remember, carbohydrates can be attached to lipids or they can be attached to proteins. This makes glycolipids and glycoproteins, and they can be on the surface of any cell. Okay, so they would be attached to or part of the plasma membrane. Well, you guys all have blood, right? So there's what's called the ABO blood system. So some of you may know your blood type. But it's the blood type, or it's the carbohydrates on the surface of the protein. Let's look at this. We've got, these are the different carbohydrates. Those are shorthand names for that, for them. They're just basically long, polysaccharides, which are actually attached to a protein on the surface of the cell, right? So these are blood cells. So you can see that the O type has this right here, but then A has this additional part attached to it, and then B has a different additional part attached to it. And those individuals with type AB blood have both of these. Okay, so we're going to talk about the genetics of that later on in the semester. However, this shows how important the carbohydrates are, which are attached to your blood cells. Now, in terms of the membranes and the synthesis of a membrane, it just kind of basically form. But in terms of the overall membrane of the cell, it can be synthesized during the process leading to vesicles. Okay, so remember, you have the Golgi apparatus, and then you have transport vesicles, and inside some of these vesicles, we can see that there are transmembrane proteins. Some of them are transmembrane glycoproteins, and then some of them are just proteins that are soluble. They're not attached to the membrane, or they're going to go through modifications through the Golgi apparatus after the ER. They could directly come from the ER as well. And then, so they're going to get transported through this system in the cell, and then these vesicles will fuse with the plasma membrane, and this creates more plasma membrane. So this kind of builds the cell. And then it actually adds, this is the way proteins are added to the surface of the plasma membrane. So you can see we've got proteins attached to, look, we've got a glycolipids, and then we've got a transmembrane protein with carbohydrates attached to it. And then you can see the vesicle is actually releasing proteins as well. So those are proteins that are secreted from the cell. Right. So in terms of its structure, the structure of the membrane results in this selective permeability. So we're going to kind of just review this, and then we'll move into transport. So the cell must exchange things. So it sends out waste and it secretes proteins, and then it takes in things. So it must be regulated in some way. You can't just have free flow of all kinds of things. There might be toxic things that might enter the cell, and you want to keep that from happening, or at least try to, anyway. So this could be, analogous to traffic, right? So you want to kind of control traffic on the highways. You don't want too much traffic. So you've got stoplights, right? So I guess a protein could serve as a stoplight, or a green light if it's something that is allowed into the cell. Now, the plasma membrane itself has its own way of regulating. However, it's not as selective. Now, the plasma membrane itself can allow molecules in and out of the cell without the help of a protein or a channel. So usually those things are very small and hydrophobic. Hydrophilic molecules must have a protein that is that helps it across the membrane, because remember, the membrane is mostly hydrophobic. So it's not going to readily interact with a hydrophilic molecule. All right, so transmembrane proteins are meant to allow the transport of larger molecules and also hydrophilic molecules across the membrane. So this can so I kind of talked about the membrane structure and the function of the membrane, and of course, the fluidity of the membrane and things that can affect the fluidity of membrane. The next segment will be all the transport stuff. Thank you for listening.